Okay, so this is kind of an overview of the material that we talked about in Lab 6, and we're going to kind of take a trip through the immune system as we go through these illustrations. And let's think of ourselves as looking at this from the perspective of how your body would deal with some type of pathogenic organism that might be attempting to lay waste to us, let us say. Okay, so our first line of defense is going to be the surface mechanisms that, instead of directly fighting disease as all of the other mechanisms do, these will be primarily meant to keep pathogens out. Okay, so we have the skin as one of our surface barriers. Okay, and the skin is continuous over our entire body surface, and it also has that nice layered, keratinized, stratified squamous epithelium in the epidermis that acts as a tough barrier to pathogens. And even though we primarily think of it as a barrier, it does have some ability to destroy pathogens, or at least slow them down, because of the secretions that it produces. Okay, so we have sweat, which is from eccrine sweat glands that has a low pH, so that would retard bacterial growth. And then we have sebum from the sebaceous glands that has antibacterial chemicals within it. Okay, so we do have some ability to kill pathogens off once they're on the surface of the skin, but the skin's main feature is to keep them out. Okay, and then we also have, as a surface barrier, our mucous membranes. Okay, and the mucous membranes are going to have the ability also, through their structure, to keep pathogens out, and they'll also produce secretions that would either retard bacterial growth or kill bacteria outright. A notable example there would be the stomach. Okay, so the stomach is lined by a mucous membrane, and it happens to have glands within it that produce acid. So that is going to be a place where you're going to have a lot of bacteria killed off before they have a chance to move through the rest of the digestive system. And just a little note, if you take a lot of anti-acid medications, you're actually diminishing the stomach's ability to do its job. Okay, so let's move on to kind of step 2. Let's say those surface barriers failed, and now there's a pathogen that has moved into the body. Now how are we going to deal with that? Okay, well, we have other non-specific mechanisms that can help us out. Here we have an example of a phagocyte. Okay, and so you can see the phagocytic cell here. You can see it engulfing these nice little green structures, which are probably yeast fungi. Okay, and they're going to help remove those from the area so they can't bother us. Okay, we also have NK cells. Okay, and NK cells are non-specific defensive cells that move through the body, taking care of abnormal cells that they encounter. Then we have the process of inflammation, which we spent a lot of time talking about in lecture. So we have our damaged site here. We would dilate our vessel, make it more permeable so that we can allow additional blood chemicals into the area to wall off the site, as well as deliver increased phagocytes to the area to deal with whatever pathogens have been deposited in the injury area. Okay, then we have 2 different types of chemicals that are going to be involved in helping us to have protection from disease. The first one here, they're both listed as antimicrobial proteins in your textbook. The first one is interferon. Okay, so interferon is essentially a substance that would be released from a cell that is infected by a virus. Okay, so you can see here's the representation of the virus particle here. It's entering into the cell, and this cell, because of the virus being here, is going to start to produce interferon. Okay, and the interferon is going to a neighboring cell and saying to that cell, it's over for me, I've been hijacked by this virus, but you can save yourself by protecting yourself from the virus. So the cell would then start producing protective proteins, and if the virus came to that cell, it would be able to deal with it and not become infected itself. Okay, so that's interferon. And then we also have the other antimicrobial protein of complement, and complement is based on plasma proteins, and complement proteins are always present in the plasma waiting for some reason to insert themselves into the membrane of a pathogenic organism. Okay, and so each of these little purple structures that you see is an individual complement protein, and when complement is activated, they form this structure in the membrane of a pathogen that basically creates a stable pore. Okay, and you can see that the contents of the cell are now leaking through the membrane, and so that's going to effectively take care of our pathogen for us through cell lysis. And then we have our last nonspecific mechanism. This is the only nonspecific mechanism that's systemic, and this is fever. Okay, so this poor kid here, looking pretty sad, has a fever, and the reason that the body temperature goes up is because this helps the body to fight off the pathogenic organism. When your body temperature goes up, your metabolic rate of all your cells goes up, and it also signals the body to put certain substances that bacteria need to reproduce into places where the bacteria can't get at them. Okay, so iron and zinc would be examples of metal molecules that would be hidden away in a cell so that the pathogens can't have them. Okay, and that would be signaled through the fever. Okay. All right, now if our non-specific mechanisms aren't enough, then we'd need to go to the specific mechanisms or the non-innate adaptive mechanisms for defense. Okay, and if we're using those mechanisms, then we're using lymphocytes. Okay, now all lymphocytes are going to be produced in the red bone marrow. Those lymphocytes that are going to become B cells will stay in the red bone marrow to do their initial training. Those lymphocytes that are going to become T cells will go to the thymus to do their training. Okay, so you can see through these histological images that there's good distinction between these two. The bone marrow, even though this is the red bone marrow, you can see these little openings. That's where there's adipose tissue. So even though it's red marrow, there's still a few fat cells basically in there, so that helps you to identify this tissue right away. The thymus, which we saw on the dissected cat, and we'll see again in a moment, has a lobular structure. Okay, so you can see those individual lobes. They kind of look like grapes. If you think about grapes on a stem, how you have lots of individual little components. If you were thinking about a set of grapes and you just kind of cut through randomly, how some of the grapes you'd catch kind of right in the middle and some of them you'd catch near an edge. So you'd have different sizes. Okay, so that gives you an idea of what the thymus is looking like. Each of these would be a separate lobule of the thymus, and you can see these nice partitions in between. Okay, so when our lymphocytes are initially formed in the red bone marrow, they're not going to have any surface receptors. When they go to their primary lymphoid organ, the bone marrow or the thymus, that's where they develop their receptor. Okay, and the B cell receptor is a bound antibody. That's why it has this Y shape. The T cell receptor has two parts. It has one part to bind to foreign antigen, and it has another part to bind to self-antigen. Okay, so the T cell receptor always has two separate components that look different from each other. Okay, compared to the B cell receptor, again, that has that Y-shaped structure of the bound antibody. Okay, now where are we going to find these cells after they form their receptor? They're going to leave the primary lymphoid organs, the red bone marrow and the thymus, and move out to secondary lymphoid organs. Okay, and one example of a secondary lymphoid organ is a lymph node. Okay, so this illustration is showing you basically where most of the lymph nodes are concentrated in the body. And you can see that there are some major groups. The group that you have in the axilla is referred to as the axillary set of nodes or axillary nodes. The ones that are in the neck, since they're near the cervical vertebrae, are the cervical nodes. And then the ones that are where the lower limbs attach to the trunk are right by that inguinal ligament, so they're referred to as the inguinal nodes. And you can see there are other sets of lymph nodes in the body. In general, lymph nodes are going to be concentrated if they're at the places where pathogens are most likely to be getting to the trunk. So near where your mucous membranes are, so close to your respiratory and digestive organs and your urogenital tract, as well as where the limbs attach to the body, and near our oral cavity, since we have our oral and nasal passages open to the outside, allowing lots of bacteria to come in. Okay. Now over here we have an individual lymph node, so we can see all of its structures. Okay, the vessels that you see on the curved side are the afferent lymphatic vessels. You can see the arrows coming in. That means that the fluid, the lymph, is entering the lymph node here. Okay, and on the indented side, or hilum of the node, we have the efferent lymphatic vessels. And you can see the arrow is indicating that lymph is leaving. There are always more afferent vessels than efferent vessels, because the afferent vessels are bringing the fluid in. You're letting the fluid kind of linger in here for a while, and that gives all of the nice immune cells that are in here a chance to look at it before it finally has a chance to leave the lymph node. Okay, the superficial layer of tissue is the cortex. The deeper layer of tissue in the node is the medulla, and the shell on the outside of the lymph node is the capsule. Okay, and then within the cortex you can see that there are little individual partitions and little nodules inside, and each one of those is filled with a germinal center, which is where a lot of your lymphocyte proliferation will take place if there's lymphocyte activation in this node.